Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221, and this is lecture number 7 of the series of our online lectures. Today we are going to talk about transcription in prokaryotes, elongation and termination. I would like to remind you that this is the continuation of the previous lecture, lecture number 6, in which we studied or talked about initiation of the process of transcription. Now we are moving ahead and we are going to talk about transcription in prokaryotes, elongation and termination. So the two processes of transcription are going to be described in detail in this lecture. The outline of today's lecture is we will talk about transcription elongation. We will briefly talk about RNA polymerase proofreading capabilities. We will talk about transcription termination. Then this transcription termination is of two types. One is called row independent terminator, and the other type is called row dependent terminator. So we will talk about these two different modes of transcription, that is row independent and row dependent terminator. So this is the outline. Now we move to our lecture, transcription elongation. Now RNA polymerase coenzyme moves along to transcribe the DNA sequence into a single strand RNA of the coding gene. When transcribing, the RNA polymerase interact with DNA sequence, forming transcription bubble. DNA double helix is reformed as the RNA polymerase moves forward. It means that when the RNA polymerase moves ahead and continue to transcribe the DNA, the remaining part which has already been transcribed, it starts to reform the double helix, because naturally these two DNA sequences, they are complementary to each other, and they have the capability of forming this hydrogen bonding again, and they form this hydrogen bonding again, and they are reformed in the DNA, and double helix is formed once again. So I will show this in a diagram how it works. But few RNA nucleotides newly synthesized form an RNA-DNA hybrid with an RNA molecule, and as transcription proceeds, single-strand RNA gets out of the RNA polymerase. Now here in this diagram, we will talk in detail about how this is happening. You see, this is the DNA which is being transcribed. Okay, this big portion, this is RNA polymerase. You have to remember this. Of course, in this DNA we have two strands. This one is the coding strand, this one, and the light blue is the template strand, this one. So you see, now this RNA polymerase is moving in this direction. You see the arrow. It is showing the direction of the movement of RNA polymerase. Now this is the newly formed RNA that has been transcribed from DNA. You see that once the transcription of this part has taken place, you see the reformation of the double helix of the DNA as the RNA polymerase move ahead. It continued to transcribe So here is the NTPs that are being recruited, nucleotides being added to the 3' end of the RNA. You have to remember that this is the 5' end of the newly formed RNA, and this is the 3' end. So the nucleotide are being added to the 3' end of the RNA. Here you see the DNA, it is anti-parallel. This is the 5' strand and this is the 3' strand. Here it is the 3' strand and here it is the 5' strand. As the RNA polymerase moves in this direction, the unwinding of DNA takes place. Unwinding helps the RNA to be transcribed. So overall, this diagram is explaining how the process of elongation of transcription is taking place. So you can see that we have DNA. RNA polymerase is doing the job of transcription. We have coding strand, we have template strand. We see here rewinding of the DNA that has already been transcribed. We see unwinding of the DNA taking place when the RNA polymerase moves forward. We see nucleotide being added to the 3' end of the RNA. Here is the NTPs that are added to the newly formed, and here RNA DNA hybrid region also occurs. So this some part of the RNA can form or can make this RNA DNA hybrid region. So this diagram overall summarizes the whole process of elongation and how it is taking place. So from here we move to the other step. Now the question that comes to mind is, does RNA polymerase have proofreading capabilities? Because there is a possibility that sometimes when the RNA is being formed, incorporation of some NTP can takes place, which is not correct. So then the question that comes to mind is that, does RNA polymerase have proofreading capabilities? The answer to this is RNA polymerase has proofreading capabilities 3' to 5'. This can happen by two ways. One is removing one nucleotide and adding correct one. The other is removing several nucleotides and adding new ones. So it depends on the situation. Sometimes process 1 can happen, and in other situations process 2 can happen. So RNA polymerase has proofreading capabilities from 3' to 5'. Okay, so now we move towards the last part of our lecture, that is transcription termination. Now there are specific, now the thing is, you see here that this DNA is a big molecule, it's a macromolecule. It doesn't happen in transcription that the process will continue for unlimited period of time, because the process is expensive in terms of energy, and if unnecessary unnecessary sequences will be transcribed, the whole process will be in a way futile. So the system is very smart, and it does not allow for futile process to take place. So there are specific signals for the termination of transcription. These signals they tell the process of termination to stop, and it can happen in two ways. One is called row independent terminator. This is also called as type 1 terminator. There is another type of termination that is called row dependent terminator. It is also known as type 2 terminator. So the process of termination can happen by two processes. One is called row independent terminator or type 1 terminator, and the other one is row dependent terminator or type 2 terminator. So we begin by trying to understand termination, that is row independent terminator or type 1 terminator. Sequences with the DNA code consisting of inverted repeat upstream of termination point. Here is DNA with inverted repeats. These are the some of the example of DNA with inverted repeats, and transcription of one strand will result in this RNA. Now, once this RNA is formed, it has the ability to fold to form secondary structure. You see here, stem loop in RNA immediately upstream from a run of uracils leads to termination of transcription. So folding to form secondary structure, this stem loop in RNA immediately upstream from a run of uracils leads to termination of transcription. Now RNA moves past the inverted repeats and transcribes the termination sequence. Because of the inverted repeat arrangement, RNA synthesized forms a hairpin loop structure. Hairpin loop makes the RNA polymerase slow down and eventually stops. This is how you see this loop CG. You see that the RNA polymerase is removed. Removal of RNA polymerase means that the process of transcription is terminated. This CG rich stem loop is shown here. It is the RNA that has formed is separated. The RNA polymerase is also separated. So this process overall shows that the termination of transcription has taken place. Now, this is another view of RNA polymerase cannot continue attached to DNA. RNA polymerase dissociate. You see here the self-complementary sequence. This is 5' to 3 and 3' DNA. This is 3' 5'. This is the RNA polymerase. Here is the hairpin loop, which is GC rich. This is the RNA that is formed. The RNA after the termination of transcription is separated from this replication transcription assembly. Now we got this RNA in the processes. The termination process has taken place. Now there is another type of termination that is called row dependent terminator. This is also called as type 2 terminator. Terminator is a sequence rich in C, or C rich in poor in G poor. It means that C rich means that there are a lot of C's, poor in G means that very few G. Terminator does not form a hairpin loop in this situation. Row protein binds to C-rich sequence upstream of the termination site. You see here row termination protein. The row termination protein is here. Now row termination protein moves along RNA. When it moves along RNA, row, which is a helicase, moves to the location of the RNA polymerase. Row unwinds the DNA RNA hybrid, and the RNA transcript is released. You have seen in the figure of elongation that I show you hybrid DNA RNA, and this row unwinds the DNA RNA hybrid, and the RNA transcript is released. RNA polymerase and row, they dissociate, and you see here row termination protein. It moves along here. It dissociates. This is the newly formed RNA. This is the row, and this is the RNA polymerase. This is the template DNA. So the movement of this row protein along this RNA happens, and this overall result in the process of termination. And this type of termination is called row dependent terminator. That means that this type of termination depends on the protein row. Here, this slide is showing the overall summary of how this whole process of transcription works. This is the sigma factor of RNA polymerase. This is RNA polymerase in step 1. It attaches near the it attaches the promoter region. This is the DNA that has to be transcribed. This is initiation. The RNA polymerase, along with the sigma in its holo enzyme form, along with the sigma factor, is attached. The process of elongation takes place. The RNA polymerase moves in this direction. The transcription has already begun. Elongation is taking place. The further movement of the RNA polymerase is taking place. You see here that the transcribed DNA is forming again the duplex of the hybrid. The arrow is showing the movement of the direction of RNA polymerase. Here is the process is coming towards its termination, and here this is the termination, in which the RNA that is formed is the result of overall the process of transcription. It has been formed, it is released. Of course, after termination process, the RNA polymerase is also released, and we get finally the RNA. So this slide gives us an overall idea of how this process of transcription takes place. Although this slide is very kind of complicated to understand if you don't get the background knowledge, this is why I have put this slide at the end of the lecture to try to present a summary of the process of transcription as a whole. So this was all about transcription in prokaryotes. I hope you enjoyed the lecture. If you have any questions, I will be very happy to answer. Please give me your feedback. Thank you very much for your attention.